Hello, this is a brief introduction to the open source project Life123. I'm showing the website right now, life123.science. Make sure it's .science. I'm Julian West, the founder and lead of this open source project. Life123 is about modeling quantitatively biological systems. More specifically, it has a focus on whole cell simulations, and that includes both prokaryote and eukaryote cells, and it's meant to be a combination of a software platform as well as a set of in silico experiments. The processes of diffusion, reactions, membranes, and macromolecules, among other things. It's very integrative, bringing together people from a variety of disciplines such as software engineering, data science, chemistry, and biology. And the philosophy of the project is to aim big, to be ready ahead of time for the computing advances of tomorrow. Life123 refers to one-dimensional, two- and three-dimensional simulations. And the idea is that lower dimensions pave the way to higher ones in terms of simplicity and speed. So what stage is the project at? On the main page, there's a version history with a detailed history of the project. Right now, as I'm making this video in late October, it's based on beta 16 version. It was released a few days ago. A lot of the development has taken place in 1D, some has taken place in 2D, and 3D will be imminent. Most of the work has been done with diffusion reactions, and membranes are the next big addition to the project. It's now in a beta stage. It's becoming much more stable. The main difference from one version to another is adding new features. Let's jump to seeing it in action. There is a one-click link on various pages on the website, including the front page, that starts a live version of the project on Binder. Binder is an organization somewhat similar to Google's Colab that allows one to run the notebooks in a repository on their host. So basically to see a live version of your files. And it's running, takes about 30 seconds, so I will fast forward to the end. Just say cancel here. And these are all the files and folders from the repository. Basically, this is a clone of the repository. So if you were to go to the GitHub repository and look at the files, this is exactly what you will see. The difference is that this is a live environment. So this is on a server with an environment that was created on the fly. You can start it just the same way I did. These are the core components for Life 1D, 2D, and 3D. So these are particular modules such as for chemicals and their reactions, and movies, which are logs of how the data changes during the experiment, and numerical reactions, visualization, and so on. So let's go back up here. Of primary interest to anyone who's using the notebooks would be the experiments. Generally speaking, this part is the libraries that provide the functions to create the simulations. And here in the experiments, you can create your own environment and run simulations. It does not have to be done necessarily in a Jupyter Lab environment, but this is one way to do it. For example, if we look at 2D and diffusion, the notebook, and there's also the Python file version of it. Those two are synchronized automatically. And if we start the notebook, here it is. It's live, and you can view it, including the graphics. You can rerun it, you can change it. If we go back up here, 2D, and then let's go up to experiments, 1D, likewise diffusion reaction, and so on. Let's look at one last location. Let's go under reaction. These are 1D reactions. Let's take a look at reaction 8. So this is the notebook called reaction 8. This is the corresponding Python file. As mentioned earlier, those are kept in sync automatically. And some, but not all, of the experiments have an associated HTML log file, which is automatically created when running the experiment. The best way to see it is right-click and then open a new browser tab. So this is an HTML file, including possibly graphics that were created when running the experiment. So I don't have time to give a more detailed tour, but if we go back on the website, there is a tab here called Experiments, and this is a perfect location to get a bird's eye view on all the existing experiments in 2D, 1D, and soon in 3D, grouped by category such as diffusion reaction or reactions with diffusion, with a title, a little description, And then if you click on any of them, it will show you a viewer of that notebook. So this is not a live version. Unlike what we saw earlier, this is a live version that's running on mindbinder.org, which is an organization that hosts your files, whereas this one is just straight from GitHub being passed through a viewer. So it looks the way a notebook looks when you're running it, including the graphics. But this is not a notebook that's live that can actually be run. Using the viewers here from the links on the experiments page is a great way to familiarize yourself with what the project can do and cannot do so far. And also every notebook, besides being an experiment, doubles as a tutorial on that particular experiment and on the functions being used to perform it. On the technology page, it discusses the technology stack. So it's Python with various Python libraries, and then visualization is mostly done with Plotly. There's also other libraries. JupyterLab is being used for the experiments for the notebooks, but it's not for the libraries, and it does not necessarily have to be used. In the software repository here, we can look at that, and this is a GitHub repository, and it looks a lot like what we saw before on the live page, because this is exactly what it is. This is the repo, a live version of it. Different parts of the website address different audiences. So for example, the page on diffusion is for people coming from a background in numerical methods. Once again, the goal of the project is to bring together different disciplines and people with different skill sets to create a cohesive platform. Membranes are going to be a big element that will be added to the project in the coming months for one dimension and then higher dimensions. This is an open source project, so an ongoing invitation to people who might be interested to participate. Here there's a page specifically dedicated to this, including a link to a microblogging, which is meant to become a discussion as well. And this microblogging covers a wide range of topics. Some of this will be of interest to different groups of people, anything from numerical methods to aspects of biological modeling and the interface between data science and software engineering, because this project really straddles both, and specialized areas like parallelizing computation or dynamical systems, membranes, and so on and so forth, visualization. Back on the website, we're still in the participate page. There's a section on the project needs meant to facilitate the coordination of people who might be interested. And here there's a listing broken down by general category, more specifically by skill set, of what the project needs right now, as well as what will be needed. Things like simulations or scientific computing, data visualization, user experience, quality control, high performance computing, and even outreach and research, data visualizations, operations, funding. By skill set here, if your skill set shows up in any of these items here, that is a suggestion for how, in concrete terms, you might have a way to become involved. And there is a contact page to reach me. Thanks for listening, and please do contact me or post on the discussion microblog if anything is of interest to you, and perhaps you might be curious about it or considering participating.